cell, it moves into the lymphatic system or through the lymphatic system, and if it comes in contact with a B cell that has the same antigen, or is presenting one that it recognizes, then it can release cytokines that will stimulate clonal expansion. A couple of things to keep in mind. The antigen that the B cell presents to the T cell is not the same antigen that the B cell recognized. Because the B cell internalizes that antigen and breaks it up into parts. If you think about it, a bacterial cell may have several different antigens. And so you could have the antigens for peptoglycan, and if it had a flagellum, you could have the antigen for flagella. And then if it's a gram-negative cell, there could be the antigen for the lipopolysaccharides. And then there could be antigens for sequences of DNA. So there could be antigen, all of these can serve as antigens that can stimulate the B cell, or the B cell can recognize. So a bacterium can have many antigens, or can serve to have many. A couple of other things we can say about the T cells. I said this last time. Never interact with free antigens, and they don't produce antibodies. We also mentioned, and I wrote this up there, that they recognize major histocompatibility complex molecules. That's the last time I'm going to say it. Right from now on, we're just going to say MHC. MHC molecules found on the surface of almost all cells. MHC1 molecules you'll find on the surface of somatic cells. Somatic cells are body cells. And MHC1 molecules are recognized by cytotoxic T cells. Helper cells recognize MHC molecules, MHC2 molecules, and these then are found on antigen-presenting cells. So B cells, macrophages, and dendritic cells have MHC2 molecules. Now, antigen-presenting cell. With the MHC class 1 molecules, what is being presented to the cytotoxic T cell is endogenous molecules, things that are from the inside of the cell. Okay, so it's essentially the cell sampling itself, exposing its insides to the cytotoxic T cell. If the cell has been infected by a bacterium or a virus, there's a good chance that those products are inside the cell, and so that they can then be presented to a cytotoxic T cell. So if a cytotoxic T cell comes along and finds a cell, a body cell, that is presenting something that cytotoxic T cell happens to recognize as bacterial or viral or abnormal, it can release cytokines that target the cell and cause the cell to undergo apoptosis, cause the cell to essentially undergo death, death by destroying itself. Essentially what happens inside the cell is the lysosomes that have all the digestive enzymes start releasing the digestive enzymes from the inside. So the micro-digested cell, preventing any viruses or bacteria stuff from being released into the environment. Cytotoxic T cells essentially go around sampling our cells for the presence of infection. Helper T cells then, we're looking at antigen-presenting cells, then these are antigens that have come from outside the cell, exogenous, if you will. These, think about what's being presented in this case. Again, B cells, macrophages, dendritic cells, what they are presenting are antigens that they've collected from outside, stuff that's in the body but they're not inside the cells. So they're collecting things that are exogenous or outside the cell to present to the T cells. And as you might expect, there are actually two classes of T cells, T helper cells. T helper 1 cells and T helper 2 cells identify specific types of MHC molecules. T helper 1 cells identify macrophages. T helper 2 cells read or recognize B cells. And of course, they both recognize dendritic cells.
So we have to know if the 2 goes with that and the 1 goes with the other one?
That's pretty much the same information I have over here.
So you want us to know what goes with?
Mm-hmm. Okay. Let's turn our attention to one more type of white B cell we haven't really talked much about. It's also a lymphocyte. It's the natural killer cells. So these guys are a little bit different than the other lymphocytes that we've been talking about. They lack antigen-specific receptors. Let me write that up here. So what that means, if we think about it, B cells, all B cells have a specific receptor on the surface that they bind to a specific antigen with. All T cells do the same thing. Natural killer cells don't have surface receptors that recognize specific antigens. So what are they recognizing? How do they know which cells to destroy? Well, we've talked about this already. These guys are involved in what is known as our antibody-dependent cellular cytotoxicity. I used that term already before. Antibody-dependent cellular cytotoxicity means that the natural killer cells are actually recognizing antibodies. They're not recognizing a specific antigen. If you remember our discussion about antibodies, that FC region is highly conserved. It's similar among the classes, and so we don't have to have, a different cell to recognize each different antigen that might be generated. So now, if you think about what we're talking about here, a long, big worm or some other kind of organism gets studded with these antibodies. And so now, the natural killer cell coming along recognizes the antibody. And so it releases cytokines that effectively destroy the microbe or the organism, usually by punching holes into the cell membranes, causing disruption of the cell membranes. This can also work to kill self cells that have become infected. So if there is a cell that has become infected with a virus or a bacterium, and antibodies are binding to that cell, the natural killer cell can bind to it, release cytokines, and destroy those cells. If a cell is lacking an MHC molecule, natural killer cells will destroy it. Sometimes an infection, the cell becomes infected, the microbe causing the infection changes the surface molecules so that the MHC molecules maybe aren't there. Great strategy, because now the T cytotoxic cell can't sample that cell, doesn't know it's been infected. But the natural killer cells can come along, recognize the anomaly, and destroy the cell. Our cancer cells can be destroyed by either of them as well, depending upon the anomaly that's being presented. But both cytotoxic T cells and natural killer cells have the ability to recognize a cancer cell and destroy it. You have cells that are becoming cancerous all the time. Even as you sit there right now, you have cells that are becoming cancerous. Fortunately for most of us, while we're young, our immune system is able to recognize that and destroy those cells before they have a chance for a tumor to develop or something like that. As you get older though, again, your immune system slows down and your ability to respond to those cells that are becoming cancerous is reduced, and so we see cancer develop in the older ages. If you live long enough, you'll die of cancer. You should hope to. I mean, really, if you think about it. That means you've lived a very long life. A couple of other things that we want to talk about here. This notion that we have 100 million different B cells and 100 million different T cells. Now, how do we come about having such a wide variety of cells, each of them with proteins on the surface that are specific to antigens? When you look at our genome, we only have about 25,000 genes. So it seems like it's impossible that we would have such a variety. Except for when we look at our genome, what we discovered is that there are sections of genes that are responsible for the coding of our B cells and T cells. So actually there are going to be several sections like this. So these are each sections of DNA, each coding for a specific protein, and there are several of these known here as the variable region. In fact, there are 65 different genes that make up the variable region. And then there's another region known as the diversity region. And there are 27 of those. And then finally, there's another section of DNA called the joining region. And there are 6 genes that make up this section. Now, when B cells are being formed, what happens is, to increase the variability, we are able to take any one from each of these 3. So if you have 65 variable regions, one B cell might get variable region 2, might get diversity region 7, might get joining region 3. What happens is that these regions that aren't being used drop out, and you get the DNA segments to come together to form what will be eventually a specific protein that will be recognized, or be able to recognize only one type of molecule out there. So this is if you think about what we're talking about here, if this is our antibody that we're developing, this here is responsible for this part of the antibody, the part that contains the active site for the antigen. There is also a third region over here that is involved in antibody production, but it's highly conserved. It gets added on here, and it is responsible for the FC region. Okay. We don't need this to be as variable. So because of this, because it's basically, if you want to think about it, it's having many different items in your wardrobe that you can mix and match. And so you can create several different outfits simply from choosing, different pair of slacks, different shirt, different tie. I'm sorry?
Well, the variable would be the FAB Both sides, right?
I'm saying, no, yeah, once all of this comes together to form this sequence of genes, that is what's going to create our FC region. So it's not just the variable region. You have to have all three together.
Oh.
Okay? All three together are responsible for creating this variable active site.
Is that just one arm of that, or is it the FAD region both arms?
Yes. So,
but you just circled the one on the left side there.
well,
I'm just clarifying. 'Cause I don't want
you've got this happening on both
Okay,
sides,
so it's-
and how they get joined can vary as well. So that can add some additional variability to our antibodies. One more thing. So we have to be concerned with our immune cells that they don't attack our own cells. I mean, it's okay if they're killing bacteria and cancer cells and things like that, but when they start attacking healthy cells, well, that creates an issue for us. So our body has a system set up that is designed to prevent that from happening.